Hi guys, let's go over week 4's stuff today. We're going to talk about kidney, and we're going to start the R2A system, review on all this stuff. Of course, it's not super in depth, but just kind of get you back up to speed. Kind of key stuff that you need to know for boards. All right, off we go. There are some birds. That's probably my best shot ever. Whitetail kite. That is in a contest right now. Fingers crossed for that one. There is Frank, the probably 3-month-old gray fox, hanging out. There is the fiasco that happens when the word gets out about interesting stuff. There are 5 whitetail kite babies this summer. And yeah, just ridiculous. The word gets out. That's why we don't ever disclose the location of nests, because this happens. And that's Randy. Randy's very curious. He's very young here, probably 2.5, 3 months. Yeah, I'd leave my gear unattended, and he's already snooping around, and very curious little gray fox. And there is a bald eagle. Another one I entered in a contest. Nothing. Not a winner, but I really like that kind of the fall look. All right, let's get into our review now. So Nephron. Here we go. And yes, some of you have emailed me. There will be some bird questions, very easy bird questions on the test, which will be your extra credit. That's kind of the curve, right? You guys know how I work. All right, Nephron review. Let's talk about it. So kind of a cool picture of a kidney without all the stroma. This is just, what is this called? The important stuff of any organ. Parenchyma. This would be the parenchyma of the kidney. We can see. Yeah, pretty cool. There's 2 of them. Hopefully, there's 2 or we're in trouble. They are derived from embryologically intermediate mesoderm, specifically from something called the nephrogenic cord. Their big job is to filter blood of almost everything. Almost everything that is in the blood plasma gets pushed inside of Bowman's space, and therefore it's said to be filtered. So lots of water, lots of you name it. Some things that are not filtered are big proteins like albumin. Albumin actually can make it into Bowman's space, but it's super negatively charged. So size-wise, it could even get in, but it can't, because negative and negative repel. Podocyte foot processes are very negative, and so is the albumin. Blood cells, red blood cells, white blood cells, platelets, too big to get filtered, so they stay in the blood. Yeah, we said that. There's 2 of them. They cannot regenerate. If you injure your kidney from, injury or disease, they can't regenerate like the liver can. So, yeah, you want to take care of your kidneys. The functional unit, of course, is the nephron. Each kidney has 1 million nephrons. So lots of nephrons. You can't see them in this picture, but the nephrons and collecting ducts live in these regions right here called pyramids, which is a region of the kidney called the medulla, as we'll see here. Actually, let's get into the 4 main parts right now. Very cool picture, going from outside to inside. A little cold starting, sorry about that. Not going to have time to edit these little things. Outside to inside, we have a capsule, which is like the skin on the outside. Very thick, very fibrous, for protection purposes. Other than that, it doesn't do anything. Then we have the cortex, which this is this region here. Okay, don't forget that the cortex also extends down, strangely enough, in the medulla. Okay, so those are called the renal columns. They don't contain any of the functional unit, but they do contain not the key blood vessels for keeping the gradient going, the osmotic gradient going, just regular blood vessels here. Great. The cortex which is up here, that contains renal corpuscles and the distal and proximal convoluted tubules of both types of nephrons live in the cortex. Great. The medulla, now we're getting down into this region, and the medulla again is penetrated by parts of the cortex, which is strange, but these renal columns really don't do that much with regard to renal physiology anyway. So the medulla contains these structures that look like pyramids. So what's inside? So the author was very cool here. He or she drew a nephron inside here. So remember, this is big, right? It's way too big. There's 1 million of these stuffed into this kidney. But we can see there's a loop of Henle here. There's Bowman's capsule there. And there's all the proximal and distal convoluted tubules that live up in the cortex. One of the few drawings that is actually anatomically correct. A lot of junk online on Google. So this is a good one. Great. So you need to know what the medulla contains. You can see the Gillard question coming, can't you? The medulla contains all of the following except for blank. So it doesn't contain Bowman's capsule, but it contains the loop of Henle. It contains all the collecting ducts. There's fun fact, there's about 12 pyramids per kidney. And each one opens into a, what's this called? Right there. Tip of my pointer. Quick. Oh, it tells you right there. Minor calyx. These two minor calyces drain into a major calyx. The major calyx drains into the renal pelvis. The renal pelvis, which has urine by this point, not tubular fluid, not filtrate urine, that drains into the ureter, which goes down to the bladder. Yep, so renal pelvis collects urine from the major calyces. Here's the urine path. So we have collecting duct. Then we go the renal papillae. Sometimes it's called Bellini's duct, I've heard it called. Renal papillae of Bellini. So boards love that question. Make sure you know what that is. What does it do? It's the junction between the collecting duct, or you could say it's the junction between the pyramids and the minor calyces, minor calyx. Okay, so there's the collecting duct, goes to the renal papillae, through the minor calyx, and renal pelvis. Make sure you know that. Another fun fact, right? You know how I like to take these little tidbits here and make questions out of them. With regard to pyramids, the pyramids, the apex, the point of the pyramid, that's the apex, this is the base, right? It's a triangle, apex base. Apex points toward the minor calyx and toward the major calyx. The base points toward the cortex. Great. Two different types of nephron. We have, and what's the nephron again? Claim to fame? Functional unit. Functional unit of the kidney. By the way, is the kidney an endocrine gland? It is erythropoietin, right? It is an endocrine gland. I could have added that. Anyway, two different types of nephron. We have cortical and we have juxtamedullary. Both of these, as shown in this picture, empty into and share a collecting duct. Collecting duct has these cortical parts, outer medullary part, inner medullary part. I didn't put that, but next time I will put that down. 3 parts of the collecting duct. Great. Cortical nephrons are, surprise, surprise, in the cortex, or most of them are. And then the juxtamedullary, even though Bowman's capsule and the proximal and distal convoluted tubule are still in the cortex, they have very long loops of Henle that extend way down into the medulla. We've got slides coming on that. Let's talk about them more. So, cooler slide. Cortical nephron. These are the workhorses. These are the ones that exchange ions. They don't have a whole bunch to do with making the deep medulla super hyper osmotic. That's not their job. Their job is to exchange sodium, potassium, hydrogen, calcium. That's what they do. They don't concentrate the urine. It's the loop of Henle. It has one, but it barely goes into the medulla. There's pericapillary tubules. I didn't get that deep this time around. Probably next time I will. Remember, this is a brand new class for me, and I just got word that I'm getting this without any time to really prepare. So anyway, peritubular capillaries surround them. The vasa recta, which is super important in the countercurrent, doesn't surround them. Vasa recta surrounds these juxtamedullary capillaries, but by far the most prevalent. They're the workhorse. Yeah. Juxtamedullary capillaries make up about 20%. So these have a loop of Henle, and we'll go over the parts again here in a second. They have a loop of Henle that extends way, way down in the medulla. Major job, to make the deep medulla super hyper osmotic. In other words, super salty. So the deep medulla is super salty. As you move up, you have a concentration gradient where that saltiness decreases. By the time you get up here, the osmolality is equal to that of the blood, about 300 milliosmol. Down here, it's about 1200 milliosmol. I think that's per liter. Some say per kilogram. What else? So main job, as I said, to make the medulla salty or hyperosmotic. How is that done? So remember your renal physiology. There's a countercurrent multiplier and a countercurrent exchanger. Vasa recta drives this countercurrent exchanger. The loop of Henle drives the countercurrent multiplier. Great. Vasa recta surrounds these juxtamedullary capillaries, but not the cortical capillaries. Right? We said that already. Let's see. What another job is to concentrate urine, so it can pull, and that's dependent on what hormone? Antidiuretic hormone, aka vasopressin. All right, let's look at the nephron a little more. So the parts of the nephron, we have a renal corpuscle, which is up in the medulla of both types of nephrons. It's this little thing right here. Renal corpuscle contains two parts. And by the way, remember, blood flows into the afferent arteriole through the glomerulus, aka glomerular capillary. And then strangely enough, it comes out not in a venule, it comes out in an efferent arteriole. And that can go down and form the vasa recta or peritubular capillaries. So the capillary extends all the way down to here. It's a very weird setup. It's a continuous capillary, by the way, once you get into the efferent arteriole and the rest of it. Again, I didn't go that deep in this time around. Next time around, I'll slowly increase the depth of this lecture. But yeah, so renal corpuscle contains two parts. Can't you see the Gillard question? Renal corpuscle is made of the blank and the blank. So it's made of the glomerulus and Bowman's capsule, which includes Bowman's space. Glomerulus, as I said, well, kind of a weird word, lazy word. I like glomerular capillary because that's what it is, glomerular capillary. But you might just see the glomerulus and you have to know it's the glomerular capillary. So hydrostatic pressure differences take blood, fluid, and push it out of the glomerular capillary into this Bowman's capsule, more specifically into Bowman's space. And remember, it pushes it through a basement membrane, and there's podocytes, and next time I'll get deeper into that. But for now, that's all the deeper I'm going. And then the fluid, once it's in Bowman's space, it's called filtrate. And we said it contains almost everything that's in blood serum, except for the big stuff like albumin and the other blood proteins and red blood cells and white blood cells and platelets. Once that filtrate gets through Bowman's space and starts making it into this snake thing, so that's the proximal convoluted tubule, it becomes tubular fluid is the correct word for it. And it'll stay tubular fluid all the way down until it gets into the minor calyx, and then it's called urine. Let's see. Yep, we said that. Afferent arterioles, kind of weird. There's no venule here. And great. Glomerulus, everything I said. Bowman's capsule in space. Yeah, it's like an interstitium, right? There's a capillary pushes stuff out, normally at the proximal end, into the interstitium. So this Bowman's space is like an interstitium. Great. Some definitions, some words. So reabsorb, that means to pull stuff out of this tubular fluid, or even pull water out of the tubular fluid and put it into the interstitium. And it could get sucked into the blood, the artery or the capillaries at that point. But that means pulling out of this tube. Okay. GFR, so the speed at which stuff is filtered, so the speed at which water and all the ions come out, and glucose even comes out, and the speed at which the river flows is the GFR. It's the rate that stuff moves from the capillary into Bowman's space. We have the filtrate, as we said. That is the stuff in Bowman's space, and then it becomes tubular fluid. I already said all that stuff. Proximal convoluted tubule is next. So it's almost like a mistake. We push all these great nutrients, including glucose, for goodness sakes, like headed toward the toilet, right? This ultimately turns into urine and goes in the toilet. So it's like, whoops, mistake, let's pull it back in. And the proximal convoluted tubule reabsorbs almost everything. 65% of sodium, 100% of glucose. There's a list that goes on and on. Pull that up from your renal phys, because that's, boards like to ask questions about that. But almost all of that filtrate, all of the solute and water gets reabsorbed in the proximal convoluted tubule. An important thing with regard to osmolarity, it is 300 milliosmol per liter at this level. If you go way down into the medulla, it's 1200 milliosmol. That's made by the countercurrent exchange, and it's made by the countercurrent multiplier, and the countercurrent exchange just keeps it that way, so that hyperosmolaric gradient isn't washed out. Again, go back to your renal physiology, because that's important stuff. All right, so great. And the key is also the amount of solutes that are reabsorbed are matched by the amount of water that's reabsorbed here. The loop of Henle, what is that? Oh, by the way, fun fact, what is this little piece called right here? That's all actually called the straight limb of the proximal convoluted tubule. It doesn't do anything different than the regular proximal convoluted tubule, to my knowledge, but it's usually not labeled, but that's what that is. The loop of Henle contains three members. We have a thin descending limb, we have a thin ascending limb, and then the famous TAL, T-A-L, thick ascending limb of Henle, which is impermeable to water, strangely enough. I believe the ascending limb is also impermeable to water. The thin descending limb is freely permeable to water. It has aquaporins channels. All right, great. What else do we need to know about the loop of Henle? There's the members. We've already said this. Its job is to create an osmotic gradient by the two countercurrent mechanisms. Super hyperosmolar, 1200 milliosmolar per liter in the medulla, and more isotonic, meaning 300 millimeter osmol in the up in the cortex here, which matches that of the blood. So the descending limb is permeable to water, ascending limb is permeable to water, or is impermeable to water, as I said. But the key, the famous cells, I could have put them in here, that you do need to know who are the cells in here that actually do the reabsorbing from the tubular fluid. Those are the NKCC2s, NIC2, NICCC2s, or sodium potassium 2-chloride co-transporters. So they live, remember your renal fizz, they live here. They pull sodium, chloride, and potassium out of the filtrate. Water tries to follow, but it can't. So therefore, this tubular fluid, even though it's super hyperosmolaric down here, by the time it gets up here, it's actually hypoosmolaric. It's hyperosmolarity. In other words, it's water-rich, sodium poor. Down here, super salty, but up here, super water-rich. That's why water is just itching to get out of this distal convoluted tubule, especially the connecting tubule and the entire entire collecting duct. Yeah, we'll talk about those principal cells in a second. Wow, this little kidney review is going longer. Maybe I'll cut it off here after we get done with the nephron. We'll see where we're at here. Okay, great. Everything we said. Some more definitions. Reabsorbers pull stuff from the filtrate. Stuff is ions, sodium, potassium, hydrogen ion, calcium bicarbonate. That's pulling it out of the filtrate or tubular fluid. That should really be tubular fluid back inside the cell, and then it's exported into the interstitium and gets into the blood. GFR, we already said, that's the glomerular filtration rate. That is the rate of filtration from the glomerulus into Bowman's space, like the flow of river. Filtrate is the stuff, we said this already, right? Tubular fluid, yeah, we said that. Distal convoluted tubules, it's the proximal part of the convoluted tubules. Really the claim to fame, and this isn't really true, it's kind of arguable where these are, but part of the R2A system, which is where we're headed, contains these special macula densa cells, which also have the sodium potassium 2-chloride channels as well. So we'll look at that. That's the only two places I know of where those two types of channels are. But yeah, the big claim to fame with the distal convoluted tubule is that that's where calcium is reabsorbed. That's where parathyroid hormone, PTH, has its effects to reabsorb calcium from the filtrate. So that's kind of the claim to fame. It is also impermeable to water, just like the tall is. Connecting tubule, sometimes called the distal convoluted tubule, or the late distal convoluted tubule, connecting tubule, that's where the magical principal cells start. So, very cool. It can't, again, impermeable to water, doesn't have aquaporin channels unless ADH is around. And what's the AKAs for ADH? Because you know I interchange them like crazy. I've seen both AKAs. I've seen all three AKAs on boards before. So antidiuretic hormone, ADH, vasopressin, AVP, arginine vasopressin, all AKAs for ADH. So yeah, so this is where water can come out and or be left in, depending on the osmolarity of the blood and of the extracellular fluid. So principal cells are really important, but that's where they are. They're in the connecting tubules, and they're in all of the collecting duct. Great. We'll look at principal cells. Another definition, glomerular capillaries. We said this already. Glomerulus, yeah. That's capillary tuft, filters blood into Bowman space. I already said that. All right, let's do the principal cell. So remembering the principal cells. So we just said where they are. They're found in all three. They're found in the connecting tubule, but in all three regions of the collecting duct. They have, just like any cell, they have two sides. Let's review the sides, because the question, if you don't know the difference between basolateral and apical, you won't even be able to answer the question. It's just assumed you know that. So here's a principal cell. They make up those tubules. They make up the walls. So they're epithelial cells that make up the walls of the tubules, connected together by strong, tight junctions. Stuff has sometimes a hard time sneaking between them. But the apical surface, the AKA will tell you, it's the luminal surface. This is the one that's in contact with the tubular fluid, or the stuff that's going to become urine. The basolateral surface is the stuff that's in contact with the interstitium. And the capillary is not drawn here, but the capillary would be hanging out over here. Okay, so make sure you know those things. You can't even come close to answering the question if you don't know what that. Like, you should know the ENaC channels are on the apical surface. ROM-K channels on the apical surface. The sodium-potassium pumps are on the basolateral surface. See how that works? So key players still. So aquaporins. So these are the big players within a principal cell. We have aquaporins channels. They are, where are they? They're not shown. They're still within their vesicle. There's an aquaporins vesicle that contains them. Remember who stimulates them to be implanted. Not aldosterone, antidiuretic hormone. Antidiuretic hormone will cause these to be inserted in here, and there's your aquaporins channels. But that only happens if we're a little dehydrated, we have our blood got a little salty, we need to dilute it. So that's how that works. Don't think I went that deep into this review, but again, I can't reteach you physiology in one lecture, so just hitting super high yield stuff. So we have ENaC channels, sodium epithelial channels. You're supposed to just call those ENaCs, but just sounds kind of weird to me, so I call them ENaC channels. We have ROM-K channels, renal outer medullary potassium channels. Their job is to move potassium out of the cell. As sodium comes in, potassium goes out. Who stimulates that? What hormone? No, not vasopressin. Aldosterone. We have some important receptors, mineralocorticoid receptors. Remember, they're cytosolic. They float around the cytosol. Who do they bind? Well, it tells you mineralocorticoid. Who's the mineralocorticoid? Aldosterone. And then we have V2 receptors on the outside of the principal cell. They bind, who do they bind? It stands for vasopressin, aka ADH. So they bind ADH. They use a second messenger system. MR and aldosterone don't. Great. Let's see. Yep, so I said down there. So antidiuretic hormone binds them. Aldosterone binds them. Great. Let's just, without going crazy into the weeds, let's just remember this. We just had this in endo, so this should be easy questions for you. You've had it several times. Cumulative final, you've had it, so should be easy. So there's a principal cell. The alpha intercalated cell, I didn't put it in here. Next time I will, but that's right next door to it. So aldosterone binds or directly stimulates principal cells and alpha intercalated cells to do these things. Remember I said day one, even back in systemic physiology, remember what aldosterone does. Because then hyperaldosteronism, hypoaldosteronism, there's so many things from just remembering, so many pathologies you can get just from remembering this. So aldosterone causes the reabsorption of salt. Water and chloride will follow. Water will come secondary to ADH secretion. Salty blood immediately stimulates the release of vasopressin, which binds there. But aldosterone we're talking about. So what does it do? By itself, reabsorption of salt through ENaC channels. It also causes the excretion of potassium through ROMK channels. Okay, also not shown here, the excretion of hydrogen ion. And really excretion, if you want to get technical, there should be secretion, not excretion. Excretion is a urine term. Secretion is what we're really talking about, but I've seen boards not really distinguish between them like they should. So next time I'll change that, but they're almost AKAs for board purposes. Here's the little fun fact. Aldosterone causes the direct absorption, reabsorption of salt, and the indirect reabsorption of water through ADH from the principal cell. Let's see. Reabsorbed sodium then will get into the bloodstream, so let's follow it. So aldosterone comes. Why am I bringing this up now? Because we're getting ready for R2A system. R2A system stimulates the release of aldosterone, right? Angiotensin 2 does. So it's a steroid hormone, so it can go right through the plasma membrane, binds to MR receptor. Those two go in, and through the central dogma, they stimulate the creation of three things: ENaC channels, ROMK channels, and extra sodium potassium ATPases. There's always some of these around. There's always some ROMK channels around as well, but you're stimulating extra ones of these. So sodium comes into the cell, goes out of the cell through sodium potassium pumps, gets into the extracellular fluid, or gets into the interstitium, gets sucked up in the distal capillary. The magnocellular neurons sense the salty blood and they release vasopressin. Here comes vasopressin, and it can't go in and directly cause anything to happen, but through a second messenger system, it stimulates the implantation of aquaporins 2 channels, and therefore water can just shoot right on through into the cell. And there's aquaporins 3 and 4 channels, ubiquitous in the basolateral surface. So basically salt comes in and water chases it. Chloride can sneak between the sides of cells and chase the salt as well. It's got several ways to get in, but there you go. So there's like a 2-hour lecture condensed into one. This is board review class though, right? You're supposed to know this stuff already. Everything I just said right there. So the weird thing about aldosterone is the actual hormone goes into the nucleus and binds to genes that secrete all these things, or that create through the central dogma. What does that mean through the central dogma? Well, they stimulate RNA polymerase to make a messenger RNA. Messenger RNA swims out, finds a ribosome, and that's where these proteins are made. Great. And let's see. Yep, everything we said. Once in the nucleus, the complex binds. Everything I just said binds to chromosome gene that makes ENaC channels, makes sodium, potassium, ROMK, and hydrogen potassium pumps, as well as proton pumps, also called, what are those called? Hydrogen ATPases, aka proton pumps. Okay, are we good? Again, you should be able to handle this stuff. Okay, let's get this back. I've been showing you this since second quarter, right? It can't escape you. Remember, you go into the boards, this is what you study. There's always board questions on this slide right here. So we're going to go through this. I think, are we going to call it a day? Yeah, let's call it a day for this. It's been kind of a crazy week of finals, so this is probably good for today. So this is, what is this? that's the adrenal gland, aka suprarenal gland. So we got to know these 3 layers. What's the mnemonic for memorizing these layers? From outside to in, GFR, glomerular filtration rate. G, zona glomerulosa. F, zona fasciculata. R, zona reticularis. What's the cell name? Glomerulosa cells, reticularis, or fasciculata cells, reticular or reticular cells, or reticular cells. What do those cells secrete? Start with the G, glomerulosa cells. They secrete mineralocorticoids. Who's the king of the mineralocorticoids? Aldosterone. The board question may ask a question about mineralocorticoids. You just have to know that these two are almost AKAs for one another. Let's go to zona fasciculata. Who does that secrete? Fasciculata cells secrete the glucocorticoids. Who's the king of the glucocorticoids? Cortisol. Go deeper. GFR, reticular cells, reticularis cells, aka. Who do they secrete? The adrenal androgens. Who are the adrenal androgens? Well, the king is really DHEA, dehydroepiandrosterone, because that can be converted into a sulfonated form, or it can be converted to androstenedione. So any of these could be considered the adrenal androgens. Even testosterone. Little testosterone can be made in the adrenal gland. Can any estrogen be made in the adrenal gland? No. Just a smidgen of testosterone. All that DHEA and androstenedione are exported, and in peripheral tissue like fat and in skin. Of course, the gonads. That's where testosterone and estrogen are made. But these are the precursors. All right, great. Now, let's not forget about the chromophene cells in the adrenal cortex or medulla? Adrenal medulla. So they secrete the catecholamines. Chromophene cells of the adrenal medulla secrete catecholamines, epinephrine, norepinephrine, a little bit of dopamine maybe too. Not maybe, it does. Catecholamines are epinephrine, norepinephrine, and dopamine. Not much dopamine though. Mainly epinephrine and norepinephrine. Great, we got that down. And now who stimulates, who drives the creation of these? And what are these all called collectively? Adrenal androgens? No, that's for just the zona reticularis. Adrenal steroids. These are all called the adrenal steroids. All right, know the stimulators. So and we're headed for the R2A system. Angiotensin 2 is the main effector we know of the R2A system. We'll go over that next week. But angiotensin 2 binds to glomerulosa cells, stimulates aldosterone release. Angiotensin 2 goes into chromophene and binds to chromophene cells and stimulates catecholamines. Don't forget this one. That's important. Great. Adrenocorticotropic hormone, ACTH. That goes into the G, the F, the zona fasciculata, stimulates fasciculata cells to make cortisol. Cool. What else does ACTH do? It also binds to the GFR, the R, zona reticularis, and it stimulates them to make dehydroepiandrosterone, or DHEA, or you could just say the adrenal androgens. That's what it does. Technically, ACTH can cause a little tiny bit of aldosterone to be secreted. Some sources say up to 25%. I've seen 5%. So it's a very weak stimulator of aldosterone as well. I doubt if boards are going to differentiate that. So what stimulates aldosterone? It's by far angiotensin 2, but there's another one. Let's not, just as powerful, is increased potassium. So a little hyperkalemia occurs. That will also stimulate production of aldosterone. How come? Well, that's dangerous, right? Ventricular fibrillation can occur from hyperkalemia. So we need to get that out. We need to secrete that into the tubular fluid to get that the heck out of the bloodstream. So that's why aldosterone comes. Remember, aldosterone comes right here, goes in, binds to a mineralocorticoid receptor, goes in, and it stimulates the creation of, what are we trying to get rid of? Potassium. So it stimulates the creation of extra ROMK channels. So potassium can get out of here. There's channels on here for right here, right? That's how it gets out of the bloodstream. Sodium, potassium, ATPases. Potassium comes in like crazy and goes right out the ROMK channel. That's how we get rid of it. So you don't go into ventricular fibrillation. All right, I think that's enough for your brains this time around. I will see you next time. Remember to email me questions. Dr. Whalen is out for the quarter, so it's just you and me. See you later.